What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're actually going a little bit deeper into meiosis and talking about the stages within that type of nuclear division. So let's go along with this journey here. So before we get into it, again, some important things to remember. Humans, what we're going to use a lot as our examples, have 46 chromosomes that are arranged in 23 pairs. We get 23 chromosomes from our mothers and 23 chromosomes from our fathers. That gives us a total of 46, excuse me, I can do math. Those paired chromosomes, we call them homologous chromosomes because that prefix homo means same and logous is kind of like a ish. So I like to say that homologous means same-ish. So they're similar chromosomes but not identical chromosomes. So when we look at a homologous chromosome, they're going to have basically the same information. Let's say eye color or hair color, or if an organism has 5 fingers or 5 toes or 6, all right? So that gene is going to be within these chromosomes, and it might have some variation. So maybe the one parent has a blue eye color, the other parent has a brown eye color. Or one parent has a gene for 5 fingers and another has a gene for 6 fingers called polydactylism. So that could also be present on different chromosomes in different areas as well. So they're similar, they code for the same things, eye color, number of fingers, hair color, etc. But they might not be identical. So they might actually produce a different protein and give some variation. So this allows for different genes to be expressed, and it's why you look a little bit different than your parents. You might have some similarities with one parent over another based on some of the genes that you've inherited, but you usually have a little bit of diversity within you as well. Okay, so remember, meiosis that we talked about previously is the separation of the homologous chromosomes. We're going to separate those same-ish chromosomes into 2 different categories through 2 cell divisions. So unlike mitosis that has 1 cell division, meiosis has a double cell division. And what we produce by the end of this is something called a gamete or a sex cell. So gametes will be your sperm cells or your egg cells, and they can also be known as haploid cells. If you look at the picture up here, so a normal cell, let's say, has 2 chromosomes, 2N, and the gamete that's produced through meiosis has 1N, so it's kind of like a half cell. It has half the number of chromosomes. If this was a human, this number would be 46 up here, and the haploid cells that are produced would have 23, representing a sperm cell and an egg cell. Okay, so 46 and 23. Haploid cells down here and normal cells up here. All right, so what are our phases of meiosis? Well, we do go through kind of the cell cycle overall, and that's going to be, there's going to be some sort of interphase where the cells are just doing normal cell functions and preparing for divisions. So just like in the cell cycle, they're growing, they're producing proteins, they're getting ready to replicate. So in this case, the cell grows and it replicates DNA. So before meiosis begins, there is a growth and replication of DNA section. Okay, so cell stuff gets done. And what do I mean by cell stuff? When I say this, I mean it's making proteins, it's breaking down sugars to make ATP, it's transporting materials across its membrane, it's building its cytoskeleton, it's modifying and packaging proteins at its Golgi apparatus. Anything a normal cell would do, it's what it's doing during these interphase moments. That's between a division, that's why it's called interphase, okay? So now we kind of get a little comparison. If you look at meiosis and mitosis and you see these diagrams here, this interphase section is going to be the top where the DNA is replicated. Remember, that happens during the S phase of interphase, which we talk mostly about during mitosis, but there is a DNA replication that happens in meiosis. And that's why with these pictures, the shade gets darker of red here and it gets darker in blue here, representing that the DNA has been replicated, or we have an additional set of DNA there. So the darkness of these is representing the quantity of DNA. Okay, so you can see it starts off with a medium shade, it gets dark, right, because DNA has been replicating. You get a medium shade here, because it's about the same amount as here, quantity-wise, and then down here, it gets very light. If you notice here, it gets darker, and then it returns to its normal color, because we're just creating these 2 daughter cells that are identical duplicates to the previous cell. All right, so let's talk through meiosis, and again, it's 2 cell divisions. So the first one we're going to talk about is meiosis 1, the first set of divisions. Now, it goes through basically the same steps that mitosis goes through. There is a PMAT cycle. You'll have a cycle of PMAT for meiosis 1, and then another cycle of that for meiosis 2. but there are some key differences. And one of the key differences is the homologous chromosomes pairing up in prophase. During mitosis and other cell divisions, you get the chromosomes condensing during prophase, forming these X-like structures, but they don't make matches or pairs. In prophase 1 for meiosis, the maternal and paternal DNA, the DNA that you got from your mother and father, actually condense down and pair up next to each other. So this represents one of the 23 you got from dad and one of the 23 you got from mom. Now, they again contain the same thing, let's say eye color or hair color or something like that. So they're matched up with each other, so we know that everything in this group has those same traits, right? The same gene types that it could be. They might look different, right? I might get blue eyes from dad and brown from mom, but it's still eye color across, okay? So it does that for everything, number of fingers, all kinds of other genes that we have that make us unique. So they pair up, and the pair up is called the synapsis. That's not a question I will ever ask you, but it sometimes comes up as a word on a test or a quiz, so I want students to not be afraid of it. So if you see the word synapsis, it just means the pair up. It's just the little space in between them right here. Now, one cool thing that happens this gives us a lot of diversity. It's why you can look a little bit like, you can have some traits that maybe your grandpa had or your grandmother had that might not be present in your mom or your father. And that's because you have crossover that occurs. So the DNA inside you that came from your mom and your father when you're producing your gametes is actually being smashed together. They actually twist in sections. Maybe a whole leg can switch, or it can, like candy cane, stripe all the way down, where DNA is getting flip-flopped between the two. So you can actually see this orange DNA here and the red DNA. You got this little cross-up that happens here. So this is a crossover event. And then at the end, you get a combination of genes that's a little bit unique, right? Some of it is the normal mom's DNA with a little bit of dad DNA in there. So that might be passed on to an offspring. So you can get a mixing from your DNA history through this crossover part. It's a very unique part of meiosis instead of mitosis. All right, so continuing on, this hopefully you can tell me what phase this is. We're in metaphase because we're lined up in the middle here, but there is a key difference. Notice how before with mitosis, they all lined up in a straight line. In meiosis, they're lining up as pairs, okay? So in here, the homologous chromosomes are paired up. And you can see you had a crossover event that happened up here, a crossover event down here, and a crossover event over here. So we have some different mixing and matching that's happening. So it is different than mitosis because, again, they're lined up by homologous pairs instead of as a straight line of all 6 chromosomes vertically. So question is, how many chromosomes are in this image? It is 6, right? I have a duplicated chromosome here, 2 duplicated chromosomes for each, and I got 3 lines of those, so that makes 6 total chromosomes, okay? Now, in anaphase, again, a little bit different. Now, instead of having those X's be ripped in half like they did in mitosis, in meiosis, the X's get moved apart from each other, okay? So the homologous chromosomes are pulled apart. So this one now moves over here, and this one now moves over here. So I'll end up with X's on this side and X's on this side. Instead of mitosis before, where we ripped those X's in half, and I would have half X's on each side, now I have full X's on each side of the diagram here. Okay, so this is when you actually have that transition from what's called a diploid, so when you have that 2N number, so that's a diploid, to now a haploid or a gamete style number. So the chromosomes at this point have been divided in half, okay? So it's no longer a full-on normal body cell is what we would call them. For instance, if I took a cell out of my skin, that's gonna have 46 chromosomes. But if I take a cell that's undergoing meiosis, it's gonna drop it to 23, okay? So that's where it cuts in half. All right. So now continuing on, we do have a telophase that happens in here. So you do now get the homologous chromosomes on opposite sides of the cell. So you get 2 new nuclei that form, as well as the cell actually divides down through a kind of cytokinesis style event, okay? The sister chromatids are still attached on the chromosomes, but notice how you have the homologous pairs are now separated. The one that was here that's teal and purple is now down here. So these pairs have been separated, but there was some crossover that happened. Notice how the colors are mixed and matched in here, showing that there was a crossover that happened in prophase 1, giving a lot of diversity to the DNA. All right, now, how is this process homologous to what we've seen in mitosis? So homologous, remember, is same-ish. So there were some similarities. Hopefully you noticed we went through a kind of PMAT series, but the biggest difference is instead of dividing up chromosomes by their sister chromatids or identical copies of DNA, we divided up the homologous chromosomes or those similar pairs. So now we have kind of a separation or haploids that are occurring on both sides. All right, so mitosis would have had the X's ripped in half, as you can see in that photo on the left-hand side there, where the sister chromatids are being pulled to the opposite ends of the cell. In meiosis, I have homologous chromosomes, full X's being removed and moved to different sides of the cell. Okay, so this is just the first division within meiosis. Now, meiosis is going to undergo another division, so that's another difference it has from mitosis. It has a 2-division cycle. So let's start looking at that second division. Okay, this one, if you look at the picture, should look kind of familiar, right? We have a prophase, this condensation of DNA in the nucleus, which is pretty much already done for the cell from the telophase 1. So prophase 2 is actually very easy for the cell to do. It's already set up that way. Notice how it kind of now lines up those in a nice orderly vertical row, kind of like we did in mitosis. Now the X's are divided in half, where the 2 sister chromatids are split, okay? So, and then we have our telophase, our new nuclei are forming there, and then we get a division that happens of that smaller cell to produce now 4 daughter cells. So this looks identical to mitosis. The only difference is instead of, in this case, starting with 4 chromosomes here, it started with 2. It started with a haploid set instead of what's called a diploid set, okay? So the sister chromatids are what's split apart instead of those homologous chromosomes. Remember, homologous chromosomes are split apart in meiosis 1. Sister chromatids are split apart, kind of just like mitosis, in meiosis 2. Okay? So the end result here, the big idea, is that you end up with 4 daughter cells that have half the chromosomes of what you started with. So for instance, these all have 2 inside of them, so that tells me a normal body cell for whatever this organism is going to have 4 chromosomes. So that way when it reproduces, let's say this is a sperm cell or an egg cell, when it's matched up with the opposite, let's say it's a sperm and it's matched up with another egg cell, and then I'll have 2 chromosomes from this cell, the 2 chromosomes from the egg cell to make a 4 chromosome new organism overall. All right, so just highlighting again those differences, right? We have this interphase part where the DNA is replicated, we duplicate it before we can divide. Then in mitosis, we end up with clones. But meiosis, we end up with half cells. We're haploid cells that have half the amount of DNA, so that way we can get 1/2 from a mother and 1/2 from a father. All right, again, showing you the 2 pictures side by side, right? DNA is duplicated to make these sister chromatids within the X. The X's are pulled apart, and we get equal clone cells through mitosis. However, during meiosis, chromosomes are duplicated. Okay, we have those sister chromatids there. The homologous pairs are matched up. We do have a little crossover that happened here, it looks like. And then once those homologous pairs are split up, then those sister chromatids can be split apart. So every cell ends up with half. If you notice, this one has 2 chromosomes in it, and the original cell had 4. So this is the haploid cell, and this is the diploid cell. Over here, we started with 2. So that's my diploid. The starting cell is always the diploid. And then over here, we end with 2, so those numbers are equal. So we stay as a diploid, or we stay as a clone. This is going to be a body cell. These, because they're half cells, are going to be gametes or sex cells. Okay, so this is going to be your sperm or your egg cell. All right, hopefully that clears up some of your issues that you might have with meiosis. If you have any questions, feel free to reach out to me via Canvas, and have a great day.